Hello. So good evening, everyone. Welcome to the 12th week discussion on the structural biology offered by NPTEL 2023. So we are starting the session and we are in the last class, I would say, for the discussion. Tomorrow, as I had promised you, and you also asked me to collect some questions, and I also wanted to take a revision class on what we have discussed till now. That will actually help you in your exams. So for that, tomorrow we will also again meet around 6 p.m. in the evening, 6 p.m. to 7 p.m. I think the link will be shared to you. This Google Meet link will be only used, so you can all join. And yeah, and we can practice some questions. I have already collected some questions, and I will discuss with you tomorrow. Then I will share with you all the questions for your future reference, so that you can also visit back whenever you want. Okay. And also I think someone asked in mail in the discussion forum of structural biology about their admit card. So I wanted to ask you whether you have got your admit card now or you have not got. Okay, okay. So I think about the time they will, if they have not mentioned, then they will again notify you in some email or in the when you log into your portal in the Swayam. So there also I think they will mention. Means I asked one of my seniors, so he was doing an NPTEL course. He was a TA, so I asked him about this. So he said that around 10 days before they give the admit card. So if they have given you the link, then that is fine. And if they have not provided you the time, then I think they will notify you in 1 or 2 days, I think. So yes, wait for then till they notify you. Okay, so other than that, I wanted to ask, is there any doubt that you want to discuss then before going to the 12th week discussion? So if you have any doubt, you can ask me now. Do you have any doubt? Okay, okay, sure. Then we can just start discussing about the week 12. And as I said that it is the last discussion session, and so let us begin. Um, if you have gone through what the week 12 content is about, then you would have noticed that it is about drug discovery and drug design. So today we will be discussing more on that topic because the whole class, the week 12 is on drug design and drug discovery. So, is my slides visible to you in your screen? On your screen, it is visible, right? Okay, okay. Thank you. Okay, let us start then. Till now, what we have discussed is all about the how we solve these structures, what are the structure determination techniques. Then we also had looked into some means how machine learning, MD simulation is helping us in simulating the different structures and also saving us some time doing the in silico works, and from that we can actually infer useful knowledge that we can actually apply in our experimental work and do a very good research. So till now, what we have talked, all of that will be applicable for this field because this is the application of what we have discussed till now. So I think for that purpose only, sir has kept this module at the end, because everything that we have discussed is somehow linked to this application that is a very old, I would say, industry, and also it's a ever going industry in biotechnology and in the biology field, that is about drug and pharmaceuticals. So, how the drug discovery is done, and sir has tried to give you a taste. I think it's also when someone can make a course on drug discovery if someone wants. Then so compiling everything and giving a gist about this topic is, I think, sir, what he wanted to convey. So so just let us start. So first, what do you mean by a drug? Okay. So as if you have looked such videos, then he started by telling that the drugs are certain chemical compounds. If we change some chemical groups in that chemical compounds, if there is any small change in the chemistry of a molecule, and if that can make a very large impact in the biological processes or in the metabolic processes of our body, then that can be considered as a drug. So, drug is a chemical substance that affects the process of mind and or body, which is used in medication, diagnosis, prevention and treatment of diseases or any abnormal condition. So, this is the definition of what I think about a drug. So, it's a chemical substance that affects the mind or body of our body or mind, and which is actually used in medication, diagnosis, prevention and treatment of diseases. So something which is a small modification, but it is imparting a very harmful effect in our body, then we can't consider that as the drug, the sense, or the drug that we are talking about. So the drug discovery is based on only those molecules, screening out only those molecules which have a very advantageous or have a very good result in our metabolic processes. Okay, so it is somehow related to the prevention, medication, diagnosis and treatment of diseases. Okay, so again, the small change in chemistry can make a large change in the biological activity. So that is from there we were means the people were interested that how a small molecule can make such impacts in the biological process. So from that, the drug design or the drug discovery, the process started. So the drug design or drug discovery is linked with finding new medications based on knowledge of biological targets. So here, whenever you will try to design a drug, then you should have a problem that you want to solve. That you should know that what molecule you are actually interested to target. Means you should know your biological target on which you want to apply your drug and improve the biological process linked with that biological target. Okay, so the first and foremost thing before drug discovery even starts is to identify biological targets on which you can apply your drugs. Okay, so there are many problems. So I have just taken the example of what sir has used, that is about nucleoside analogs. Okay. So if you remember when we talked about the DNA replication as well as the structure of the DNA, then if you remember, then this DNA has three important components, that is this nitrogenous bases, the pentose sugar and the phosphate backbone. Among these, nitrogenous bases, they come together to form these hydrogen bonds. Okay, and during the process of replication, it's always the 3' OH group. They, through that end, the 3' OH group, the extension of one strand is occurring. Okay, new nucleotides come and the DNA polymerase will recognize only those strands where it will have a or those bases which will have a 3' OH. Okay, and that is actually important for for the replication process to take place and to get the double stranded DNA. Okay, if this 3' OH group that is present normally in any nucleoside or nucleotide, I would say, then if this 3' OH group is absent in the means I am telling about in the sugar, then there will be halt in the replication as the DNA polymerase will not be able to correctly recognize and incorporate new DNTPs and to synthesize the new strand. Okay, so that was the biological knowledge that we knew from the DNA replication process. Now, if you also have the knowledge about cancers, then you would have you would have noticed that in cancer there is uncontrolled cell division, and also the DNA synthesis is also since there is uncontrolled cell division, so a lot of DNA it needs to be synthesized, and so for that, so one of the mechanism in stopping those uncontrolled cell division, or I would say the uncontrolled cell cycle that we have in our body, then people have came up with an idea that if we can use this knowledge that we have from the basics of the molecular biology in replication to treat these diseases. So what they have done is that they have taken the nucleotides which has this pentose sugar and this which have this 3' OH. It is only and what they have done is that at the 3' OH they have removed that 3' OH, and they term this as dideoxycytidine. If you also remember, or you can correlate after when I say that dideoxycytidine, so where we use this kind of molecules. If you remember correctly, then you will remember that in Sanger sequencing, when we were actually when we were actually trying to sequence our whole DNA strands, okay. So then what we use is DDNTPs. If you remember, so they were dideoxy NTPs. So once they will get incorporated into the newly forming strands, then the new strand synthesis will be halted. Okay. So we have used that dideoxycytidine, and that is actually used in treating various kinds of diseases where there is uncontrolled cell division and there is some problem with the DNA synthesis and the cell cycle. Okay, where we want to terminate the DNA synthesis since it is leading to certain diseases. So those modification in the nucleoside are known as these nucleoside analogs. So other than this, there is also L-nucleoside analog. So it has been observed that if it is an L-nucleoside analog, then the DNA polymerase cannot easily recognize this analog, and cannot the replication process is halted. But the actual mechanism behind this is still under research. So if you have looked into such slide, then he had proposed one mechanism which he I think performed in his PhD. So you can go into his slides and look if you have more interest. And also some people are using open chain analog where you are actually removing the pentose ring, and there is only an open chain. So this is a kind of medicine acyclovir if you have heard. Okay, so there you actually use this open chain analog. So this is an example where you can just make a small change in the nucleotides, and you can get a very huge impact in the ultimate process that we are interested in. Okay, so from this we will build up our knowledge of drug discovery. Another important thing that you should know while going to the field of drug discovery is what is a hit and what is a lead compound. So a hit is any compound that has binding activity to the biological target. So hit is any compound which can actually bind to a biological target. But whether that means that it will impart some function, it is not always that case. The heat is any compound which can actually bind to your biological target. For example, if you are targeting some receptor, then there are many compounds which can actually bind to your receptor, but it does not mean that it can actually modify the cytosolic, means it can transmit a signal through the receptor so that there will be some cytosolic signaling from the receptor and you will get the biological process. So the compounds which are actually able to do so, that is a lead, the compounds which will have a pharmacological activity, but still you need to optimize it by various clinical studies, and so that you are following all the safety protocols that needs to be done before it is ultimately converted to a drug. But that molecule which will have a actually pharmacological activity, those hits which will have a pharmacological activity are called as lead compounds. Okay, so this should be clear. Now, what are the different types of drug discovery? So, or drug design, I would say. One is traditional drug discovery, another one is rational drug discovery. So, traditional drug discovery, from the name itself, it suggests that it is somehow linking to our traditions. Means it's if you think that the Ayurveda, it's the best example is Ayurveda, where it is a very rich science that I think we are not able to understand yet. Many informations are not yet, means not, they are not documented in such a way that that's why I think there is a gap in this field. But people are also working in this field to come out with newer therapies which has been discovered previously by the saints, I would say, but they didn't know the scientific relevance. But now what we want is that to derive those practices which they used to perform and try to find the science, the logic behind it. So that is the traditional drug discovery about. Whereas the rational drug discovery, it's a new field of drug discovery where we are mostly utilizing the resources, or I would say the structural biology techniques, all the computational techniques that we have discussed. So all is utilized in this rational drug discovery to come out with a novel drug candidate, and there are also many screening techniques that we will be discussing. So in nutshell, if you want to differentiate between them, then traditional drug discovery involves the origin of drug discovery, means it's the origin or the from the start, where when the drug discovery started, how this drug designing is going on, that is under the traditional drug discovery. Rational drug design begins with a hypothesis that modulation of a specific biological target may have therapeutic value. So, in rational drug discovery, we do not start the process blindly. We have a rationale behind that, that why we are modifying this chemical compound in such a chemical compound which will actually impart certain benefits to to treat our certain diseases. So we know the biological target and all these what we want to target and what process we want to modify, all this information we had hypothesized previously, and then we start the drug discovery process. So traditional discovery is evolved in natural sources or certain accidental events occurred which has given us means something, I would say that they were accidental discoveries, but in science you would find that much of the discoveries which were groundbreaking discoveries, they came from certain accidental discoveries only. Okay, so they were from natural sources, and whereas the rational drug discoveries is more targeted and it's not an accidental discovery, it's more targeted and more systematic. The traditional discovery is based on trial and error method, whereas in rational drug discovery, as I said, that we had the previous knowledge, so we are more equipped that it is not a trial and error method. Otherwise, it would take a lot of resources, money, and time. Okay, so that's why we have moved towards more towards the rational drug discovery approach. Now, in a nutshell, in one slide, if I want to put all the topics that Sir has discussed, so I have just compiled them and put them in one slide so that it will be easy for you to read in future. So, its traditional drug discovery is further divided into certain categories, and they are random screening, trial and error method, ethnopharmacology method, classical pharmacology, or by serendipitous discovery. Okay? So random screening. From the name itself, it suggests the screening of certain selected class of molecular compounds like alkaloids, flavonoids, any synthetic library collection. Okay, so we are coming with a means when this random screening were done, so they came with a series of compounds. I think which they know that they had some benefits, and they wanted to see that with those compounds whether they could treat a particular disease or not. Okay, so they were doing that randomly, means they didn't know any specificity what biological target to they have to actually pinpoint and target. They didn't know, they were just trying the try means in the trial and error method we will come. So they are screening of randomly selected traditionally identified medicinal plants. They were taking certain medicinal plants and they were randomly screening their effect in certain diseases. So that's come under the random screening. Then trial and error method. So they took different resources that were present in the nature, that is various roots, leaves, barks that could be used for medicinal purposes. For example, cinchona bark contains quinine, and if you know that this quinine is actually used for treating malaria. So see, means it's not like that the traditional drug discovery has not taught us anything. Means it is one of the type of drug discovery which I think we have not understood very well. But there are many important contributions of this field that we are still using. Like for the example is like the quinine that is actually used to treat malaria. Ethnopharmacology approach is actually linked to the ethnopharmacology, means at or a particular habitat in which they are more related to the culture and the specific cultural groups that were there in India. We have so many cultural groups, so you would have noticed that in your home also, if you are falling sick, then sometimes your mother or grandmother comes up with a home medicine. I would say that they would suggest something and it will certainly work. But how will you means then you would ask then how it is working. It is based on their experience, and that is also a type of traditional followings that they have been taught before. That means, for example, for cooling your body, means in Bengali we say dabe jol khao. So means since Kolkata is now too hot, so you should also drink dab. So yes, in that way means they have evolved in means the drug discovery has also evolved with the cultural groups. Okay, so those practices are under this ethnopharmacology approach. Okay, then classical pharmacology, which is function based approach, where we actually know the functions here. So the prevention of experimentally induced seizure, lowering of blood sugar, suppression of inflammatory response, means here we do not, they didn't knew the biological target which one to target, but they knew what they should, what these molecules that they are using or those techniques that they are using for treating a disease. So what their functions, what are the functions it need to perform, that they know, and then they are designing some drugs that it is forming and falling under classical pharmacology. And if it is serendipitous discovery, then the best example that Sir has also discussed is by the discovery of penicillin by Alexander Fleming. Okay, so for which Fleming, Florey, and Chen, all of them, they got Nobel Prize in physiology. So if you know the story that he was culturing some bacterial plates, and there suddenly means after coming from his vacation, he found that there is a mold grown in his hello, am I audible? Hello. Hello, am I audible? Hello, 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 am I audible? Yeah, I had shared, but I think in between the connection there was some problem with the connection. So that's why I was asking whether I am audible or not. Okay, so I was audible, right? Means till which part means whatever I have discussed, you were able to hear, or okay, okay. So just we were discussing. So I think my slides are visible now. I have shared my screen as well, so it is visible, right? Okay, okay. Okay, okay, okay. Thank you. So yes, so I was talking about the serendipitous discovery of penicillin. So where means I was just telling you the story behind it. So there was these bacterial plates that Alexander Fleming were using for was using for the bacterial culture, and after coming from a vacation, he suddenly discovered that there are certain molds which are formed in his culture plates where the colonies of those bacteria were not there. Okay, so that mold that he found, so from there he actually at that time that mold was actually crushed, and from that mold this penicillin discovery actually it was done. So that is about this serendipitous discovery, some accidental discoveries that happened, but that has revolutionized the field of drug discovery. So this is all about the traditional drug discovery processes. But means and also means what are the different steps involved in the traditional drug discovery? You start with a lead generation, means here your lead compounds will be the natural ligands. Okay, so which you need to screen, and means you have to go for some biochemical testing as well as the biological testing, and it is mostly based on the function, whether it is giving you a good means the appropriate function that you had thought about and you started the whole process of this traditional drug discovery. If it is not, then you actually have to iterate this process till that level to which you are getting the means function, the appropriate function of your drug. So what are the basic steps? The lead generation, biochemical testing, biological testing, synthesis, and as well as the preclinical studies and the clinical studies, which are after you are getting a biological function in a small scale, then you actually test the means this function of this, means what are the, if there is any side effects, or you for a long period of time you actually do these clinical studies before you publish your drugs into the market. Okay, But the traditional drug discovery, as I was telling, that it came with a lot of challenges. So here you need a huge investment in time and money, since you are not going into a directed approach or a systematic approach. So it can take 1 year, 2 year, and a lot of years still. If after giving so much time, you may end up with means nothing in your hand. That's a wastage, a lot of time as well as money, and it also has a high failure rate, and lack of this efficacy may have some adverse effects as well. So people have thought about these problems, and they actually came up with the solution that is known as this rational drug designing. So now let us look what is rational drug designing, and this is which we are more interested on, and we will talk in more details about this approach. Okay, so if you say that what is rational drug designing, that I have already defined, that you make some hypothesis, you know about certain information, that what is the biological problem, what is the biological target that you want to means actually address, or you want to design a novel molecule for which biological target. All this information is previously known, and then you then start the drug designing process. So again, what information you know, on the basis of that, it is divided into two types, that is structure based and ligand based. Structure based means development of molecules with desired properties for targets having a known structure and function. Here, what is the prerequisite is that you should be aware of the three-dimensional structure of and the function related to the biological target which you want to target through this drug designing approach. Okay, so that is the prerequisite for structure-based rational drug designing. For ligand-based rational drug designing, you need not means you suppose you are not being able to solve the structure of that three-dimensional biological target. So develop on then what? Then you can also do this ligand-based rational drug designing, where this development of the molecules with predefined properties for targets whose structural information may or may not be known. Okay, so here we are more concerned about the ligands, means how these ligands should behave so that it can actually attack or target its biological target whose structure may or may not be known. Okay, and in structure-based drug discovery, it involves molecular interaction calculations between each molecule to be tested and the biological target, and we can also do de novo drug designing with the help of this structure-based rational drug designing approach. Whereas in ligand based, since we don't know the structure or the three-dimensional structure of the receptor, that's why we actually put certain a set of geometrical rules of physical chemical properties. With that, we actually try to screen our ligand. Okay, so what is the overall process? We will go into more details about both these techniques in the near slides. Okay, so the overall process is given in this slide as well. The structure based drug discovery, it starts with the target structure. You do the virtual screening of your lead heat molecules, which I would say that you are not now sure that whether they will make the pharmacological activity, but then from the virtual screening you actually emit those molecules which are omit those molecules which are not giving any pharmacological activities, and you do perform molecular docking, which we will be actually discussing, and also some molecular dynamics, and ultimately you will go for structure based pharmacophore screening. Okay, and similarly for ligand based drug designing, you need a pharmacophore model, since you do not have a means a ligand based discovery drug discovery is divided into two arms, that is pharmacophore model or QSAR, 3D QSAR, that we will discuss, and then going for a ligand based screening. So this is the overall process of the that we need to carry out in structure based and ligand based rational drug design. So first is the about we will talk about the ligand based the drug designing. So as you have looked into the previous slide as well, my connection was lost, right? It's giving a lot of problem today, I think. So yeah, but you were able to hear, or I shall just continue from where I left. Yeah, means I think the connection was lost, right? Means to which part you have means you were able to listen. Um, this slide you were able to listen, right? Means we were just starting QSAR, the quantitative structure activity relationships. So from this slide, should I continue or means which part you have listened that I was asking because I think the network, the connection was gone. Okay. So let me discuss what is this QSAR. So QSAR is quantitative structure activity relationship, and this is a part of ligand-based drug designing. So here, what we are doing is that this QSR are the mathematical relationships linking the chemical structures with the biological activity using physiochemical or any other derived property as an interface. Okay, so since here we do not know the structure, we do not know the information about the structure of our biological target, we only know the biological activity that we want to address in our drug designing process, and we have certain set of ligands. So we want to develop an algorithm, the mathematical algorithm, which will connect these two, and in that process, we will actually do not overcome the means the need of the three-dimensional structure of our molecules because it's a theoretical assumption. So if we can link the chemical structures with the biological activity. So that is the prime focus of this QSAR technique. So what we are doing, we have mathematical function of chemical properties. Okay, so there are different chemical properties which we actually use them as function, and with the help of those function we are determining the biological activity. So it is like a y equals to f of x. So where this y, you can consider this, it means if you want to see it in a mathematical form, then it is y equals to f of x, where y is your biological effect, which is the outcome, and this x that which you are using. So this x are the chemical properties. Okay, so these functions, the function that you will apply on the chemical properties to get a biological effect, so they are based on certain chemical descriptors. Okay, so each chemical compound we calculate each chemical compound we calculate a series of parameters are should be present on the basis of which we actually calculate the outcome from these chemical properties. Okay, so they are known as the chemical descriptors. So there are different kind of chemical descriptors. Some I think sir has also mentioned. I am not going into details about that since I wanted to give you an overview of what QSAR is. It is an algorithm that provides a quite accurate value similar to the experimental value. So whenever you design a first, you need to come up with this model. Okay, for each biological compound or each biological problem that you are addressing, they have a different approach, different mathematical models that you need to create. Okay, Fortunately, people are working on it, and more mathematical models are coming out, and you need to choose which mathematical model is fitting your data more correctly. Okay, so once you have your mathematical model, you are starting with certain known compounds whose biological activity you know. So in that way, you are actually validating the model you are using, whether it is functioning for your purpose or not. So you start with certain molecules, for example, some toxic molecules which you know that its biological activity is very toxic, or it will cause toxicity. Okay, so with those compounds you actually start your screening, and after you think that after getting a series of toxic compounds and getting the correct results, the kind of biological activity that you want, if you get in that mathematical form, then you actually plot it, like it means you actually do this, see, molecular descriptors and response variable in mathematical term. Then you actually plot it in a graph with the help of certain statistical analysis like regression analysis, and then you want to test your model with an unknown, means a molecule. Okay, so which you want to actually know those function that you don't know about those function previously, but you are testing with a unknown molecule, like check if so often algorithms capable to predict properly the values of other chemicals which are not unknown molecules. I would say unknown molecules in the sense that they were not used to set up the model. So any other molecule whose function you definitely you need to know, but that those molecules were not being used to set up this mathematical model. Those molecules you take up, you get that molecular descriptors information, you get your response variable, and ultimately you want to check whether your mathematical model is correct or not. So in that way, you are actually screening your ligands. Okay, so that is one of the approach of ligand-based drug design. So one of the which method which can be used, one of the softwares that can be used for 3D QSAR is this COM-FA. Okay, so then we come to the second process of ligand-based drug designing, that is the pharmacophore development. So a pharmacophore is an abstract description of the molecular features that are necessary for molecular recognition of a ligand by a biological macromolecule. So here also we actually do not know the structure of our biological target, but we are making an abstract description since we know that we have previously worked with many drugs as well as their receptors. So we know what are the basic properties that should be present for our set of ligand, so that it can be able to target the type of biological target that we want to address. So that knowledge we actually acquired from different pharmacophores that has been derived previously. Okay, so it is about an abstract description of the molecular features. It is not the real description, but certain abstract description of the molecular features with which we want to actually predict that if those features are actually present in that ligand molecule, then it can easily fit our biological target. Okay, so as I have said that ligand receptor interactions are typically, it has been observed that they are either polar positive, either polar negative. There should be certain hydrophobic amino acids as well. So all these informations we have actually acquired from previous drug discoveries that has been done. Okay, so with the help of that we actually select a training set of ligands. The set of molecules should include both active and inactive molecules. Inactive molecules in the sense that will actually not give you the result. Means if to means decrease your biasness, you should also take some unrelated compounds which give, and you want to see that whether your whole development, the pharmacophore development is working or not. Then you do some conformational analysis, means you do the backbone analysis as well as the conformational analysis, where you are taking a set of ligands, we are taking a pharmacophore model, and you want to see which of them actually fits well. So with that, you actually have a, you want to minimize the energy of that process, and if it is a lower energy conformations, then we consider is it that this will be more biologically active, or it is the bioactive conformation. So we actually decrease the conformation of the ligands in such a way that it is aligning good with the pharmacophore model. Then we do molecular superimposition, that I was talking, that similar biosteric functional groups common to all molecules in the set might be fitted. Means there are certain common molecules, means common functional groups that you will find after aligning with one functional group with another, and you actually take the best fitted ligand out of all the ligands that you have selected. Then you do the abstraction, transform the superimposed molecules into an abstract representation. So here you are not getting a single molecule, right? You are taking some information from all the ligands as well as from the pharmacophore model to come into a lowest energy molecule which you need, which is an abstract. So you now need to transform this abstract into, means you need to get an abstract representation of the ligand that you want to use for your biological targeting, the biological means problem or biological target. Then you validate your pharmacophore model. A pharmacophore model is a hypothesis accounting for the observed biological activities of a set of molecules that bind to a common biological target. Okay, so here when we use this pharmacophore model is that means when we know that there are diverse molecules which can bind to a single biological target and provides its function, then this pharmacophore model is very useful. The model is only valid insofar as it is able to account that for differences in biological activity of a range of molecules. So as I discussed that you need to take a range of molecules, then you need to validate your pharmacophore model before further going into the further process of drug development. So these are the two basic processes of ligand based drug designing. Now we are going more into the structure based drug designing. In the structure based drug designing, it is also known as the computer assisted drug designing, because here we know previously the three-dimensional structure of our biological target. Okay, so we already know the structure. So if we know the structure, we know till now that whatever we have talked about. So now from the structure we can get very valuable insights that which will actually help us to design the drugs in a very specific manner. So that's why we are first utilizing the computer. So different MD simulation we do as well as docking. And if means if we are starting with from scratch, then we do de novo drug design. So that's why there are two different types of structure based drug designing, that is de novo drug designing and docking based virtual screening. Okay, so what are the different steps of the structure based means drug designing? First is you need to, as we know that you need to purify your protein and you need to get the three-dimensional structure of the biological target that you are interested on. Okay, then your identification of the target binding site. Once you get the three-dimensional structure, you know, for example, you are working on an enzyme, you get the three-dimensional structure of your enzyme. Now you know what is the binding pocket of your enzyme that you should be able to visualize from the three-dimensional structure of your enzyme. Okay, so with that information, we are virtually screening of drug-like molecules from chemical databases. So there are certain chemical databases. This ACS sir has already talked about. So this is a chemical small ligand databases from there, which has some drug-like characteristics previously reported. Then starting with those molecules will actually help us in the further screening process. Okay, so we start with certain libraries of molecules which has some drug-like properties and we want to test their role in our biological target. Okay, then we perform a molecular docking and structure based virtual screening. So what is molecular docking, that we will also discuss a little bit. and then we after we get certain molecules which actually are docking well to our biological target, then we perform also the MD simulation. Means if it is an enzyme, then you may be interested that there are different states of enzyme. So which state you want to actually target, so that can actually be easily visualized with these MD simulation techniques. Then first, you do the chemical synthesis of top heat molecules, means then it's experimental based. Means after you got this information, you now narrow down your pool to certain compounds which is specifically interacting with your biological target, and you think from the MD simulation that this is actually giving us certain role that we are interested in that process. So then we are taking those compounds and we are synthesizing them with many solid state synthesis can happening can be done in the lab, and with that we are actually synthesizing those compounds, and then we are doing the biological assess, and in very preclinical studies have been done, and also we need to test them in a biological model, and then we can say that if it is giving a pharmacophore activity, then we can say that with the heat that we started our process, we are now that is becoming a lead molecule, but that need to be further go to some preclinical studies before we get a drug. Okay, so that is the overall process of structure based drug design. Now we are moving to the de novo drug designing. So this kind of drug designing means information available about the target receptor but no existing leads that can interact. Okay, so means you know the structure, you have to know the structure of the three-dimensional biological target in structure-based drug discovery. Okay, that should be clear. But here you are not getting any lead compound which is actually which can interact with your biological target. So for that you need to design your lead compounds de novo from scratch. Okay, so there this de novo drug designing is playing a role. In de novo drug designing, the structure of the target should or must be known to a high resolution, and the binding to site must be well defined. Means in which site it should bind, all those parameters should be well defined before the de novo drug designing. Hypothetical interaction sites typically consisting of hydrogen bond, electrostatic and non-covalent interactions. So here also you can find some analogies with these pharmacophore development models. Okay, so there are two types of designing that you can do. One is receptor based design, another one is pharmacophore based design. Already we have talked about the pharmacophore based design. So you can have a look into this picture where you are taking the help of, you know, the biological target, what shape, means if you are seeing the shape, and then from the known compound as well as you are from different known compounds with different means you know the what is the hydrophobicity, what is the hydrophilicity, the charge interactions which can be involved in this pocket, and with the based of that you are developing your pharmacophore and selecting your ligand, or you can actually get the information about this structure and directly means construct your lead molecule with the help of this de novo drug designing, where you can actually synthesize different structures to actually improve the different lead compounds which will improve the overall drug designing process. So what are the steps of de novo drug designing? Generation of a potential primary constraints. Okay, on the basis of that you want to choose or you want to design your lead compounds. Derivation of interaction sites, you need to know what is the interaction sites, then you need to build up methods, and you need to also come up with some certain scoring function on the basis of which you can say that this is the best fit or this is the lowest energy conformation which we consider as a bioactive conformation. Then there are different search strategies which are involved inside it, and then you actually ultimately design your drug with this de novo drug designing approach. Okay. Now another is known as this docking based virtual spinning. So here what do you mean by virtual spinning? Virtual spinning is a computational technique It is used in drug discovery to search libraries of small molecules in order to identify those structures which are most likely to bind to a drug target, typically a protein receptor or enzyme. So it's a computational technique which is used in drug design, in drug discovery to search. As we know that, as I was talking, that there are a library of small molecules. From that, you need to search your lead molecule that will give a result of your choice. Means for that the purpose you started the drug discovery process. Okay, so library of small molecules in order to identify only those structures which are most likely to bind to a drug target. So here comes the information about the docking. Okay, so there can be target structure which can be unknown, which can be known. So if it is known, that we can easily, means these known structures can be done with, as we know that from the experimental techniques, as well as there are different modeling techniques that we have also discussed in our previous discussions. So on the basis of that, we have the three-dimensional structure, and with that information we can perform a docking. Okay, and if it is unknown, then the superposition and the field based similarity. So there are descriptors and this pharmacophore de novo drug, means in de novo drug designing also you need to know the target structure. So then if it is unknown, then it is more towards the ligand based drug design. Okay, and so now we are coming to docking. So docking attempts to find the best matching between two molecules. It includes finding the right key for the right lock. Okay, so means as we know that every lock has a particular key, and if you know that, if you also know about the lock and key hypothesis of the induced fit theory and the lock and key theory of the enzymes, then you will be more able to understand that every lock has a unique key that can open it. So you need to find that right key from you have the lock here is your biological target. You know what are the dimensions of the means the lock which you want to open. So the opening means you are here I am inferring to the biological function. So you need to find a right key, that is your right lead compound, which can actually interact specifically to your binding pocket of the biological target and imparts its function. So how will you do that? So that process is which the process by which we actually find this best matching is the docking. Okay. So here, if okay, so I also have the docking scheme. So where whether the two molecules interact, if so, what is the orientation that maximizes the interaction while minimizing the total energy of the complex? So what I mean is that in a docking you have a force field, just like the means your MD simulation when we talked about different force fields are there. So there are different charge distribution, different force fields are there, and you are taking your three-dimensional molecule into that force field. And if I talk about rigid body docking, where we are considering those molecules that we are docking, they are rigid bodies, which means by this I mean that they are not flexible enough, they do not have a flexible binding pocket. Means in some transcription factors that I was talking about, that there is some transient making of certain alpha helix, and then again they will denature into random coils. So those kind of flexibilities are not there. We know that this is a rigid structure, this is a binding receptor, and this is a ligand, and we are fixing one of the molecule in the space. Okay, fixing means they are in the force field, so they are aligning in a particular direction in the force field, and by not altering its position we are bringing near the ligand towards it. Okay, now we are not means doing anything with that receptor. So as the ligand approaches, we know where is the binding. We do not means we know the binding pocket where it is there, but the computer doesn't know, right? So when it comes near, it will try to interact at different binding means regions according to the lowest energy, means the del G value, they will interact and there is a scoring function, and on the basis of that, for each interaction they will calculate the free energy, the del G value. So the those which will have the lowest DLG value, that will be chosen as a best docking pose or the best docking complex, I would say. Okay, so here also we are considering the fact that lowest energy means the best biological activity in that process. Okay, so while minimizing the total energy of the complex. So this ligand is coming, it has three translational translational degrees of freedom, three rotational degrees of freedom. So on the basis of that, it will rotate itself, translate itself, there will be multiple interactions that will be going on, only the best means poses which will have the lowest del G values, they will be documented by our docking software. Okay, so this is the basic means basic outlook behind the docking, or the basic mechanism behind how this docking process works. Now, so this is the overall scheme that you need to follow. So always, if you have a three-dimensional structure, you as I said that in MD simulation also we actually remove the hydrogen and we add the hydrogens on the basis of the algorithm that particular software is following, because they can be following different charge fields, different force fields. So then on the basis of that we actually add those hydrogens. So we need the target, we download it from the PDB and we have these small molecules from certain databases, like I would show you PubChem. Okay, so from PubChem you can get a small ligand, and then in both of them, means this is a PDB format, right? But you need to convert them into the .pdbqt format. Only then you can actually dock them. Okay, so means sometimes nowadays means this process is more for, I would say, autodoc, for using autodoc. Okay, so if you have if someone has done docking, then you would have heard autodoc. So it is very useful for actually means docking of small ligands with a receptor. But I have seen that long two protein molecules which are more which are larger in size, then I think I will suggest you to go for this class pro Z dog. Then they are much more easier. Okay, but for small ligands, Autodoc can be easily used, and it's one of the renowned software for the docking. Okay, so you have this target.pdbqt, ligand.pdbqt. You need to define certain parameters on the basis of which this docking will be done, because if you the way I explained, that if you see that there will be infinite combinations to which it can interact, and that will, if it starts to compute all the means conformations and in the force field, then the problem will be that it will take a lot of time and it will take a lot of memory to run as well your means docking. So for you don't want to do that, so that's why you are specifying certain parameters. For example, you have a very big structure, okay, but you know that your binding pocket is not that much big, it's a small binding pocket, but the overall structure is very large. Then you can specify that binding pocket with a grid box that will consider that in which region you want to actually see the docking. Okay, so you need to specify your grid spacing, grid center, all these means all these parameters you need to define before you start the docking. Okay, then I will also share with you, if I have time then I will go through that, but I previously made a presentation on Autodoc, means how if someone is starting to work in Autodoc and he or she don't know how it works, then it's a step by step information through which you can, if you follow that, then you will be able to perform the docking in Autodesk. So that I have, if I have time today, that I will discuss, or otherwise I will directly share with you in your Google Drive. So from there you can actually get access to it, and you can start using Autodock if you are looking to perform Autodock in future. Okay. So after you process your files, there are different kinds of files. There will be a grid file, there will be a docking file, and after all that you prepare, then you actually run your docking. Okay, and you get the docking result. And on the basis of your docking result, it will means different softwares has different scoring functions, on the basis of which it will give the different poses, means which one is of this means high, which is the most favorable docking configurations, they will give you a list. Okay, so then you need to choose the means which actually has a correct docking confirmation. If you have previous knowledge about the structure, as well as what kind of interactions it may have happened with other members of the structure similar to your biological target, then from that also you will be able to tell that which docking structure is more means related to your experimental results. So in that way you can narrow down your means different poses or hits that will be given by a docking software. So what are the basic requirements for docking? You need a three-dimensional structure of your protein. You need a library of small molecules. You need selection of proper protein ligand complex. You also need to select a proper protein and ligand complex. Okay. So these are the basic requirements for a docking. So there are different kinds of docking. Okay. So as I was discussing with you, where we were keeping both the ligand and the receptor as rigid bodies. So that is known as rigid receptor, rigid ligand. Then there can be your receptor is rigid but your ligand is flexible. So rigid receptor, flexible ligand, flexible receptor, rigid ligand, as well as both flexible receptor, flexible ligand. These all kinds of docking can be done. But as you go downwards in this list, the complexity associated with this docking, they increase. Okay, so normally what we do in our normal means whatever we do in our drug designing, normally we use rigid receptor, rigid ligand based docking. Okay, but this can be also be done. You need higher computational power, as well as you the complexity associated with this docking will also increase. Okay, so there are certain links I have provided in the bottom, so which you can go, and as I was telling you, class pro, zdoc, so these are more towards the protein interaction. Large molecules, they can be easily docked with this software. They are available in web, they are free, so you just need to download your pdb structures. You need not to means you do not need to change into PDBQT. You just need to upload your PDB structures into this ZDoc or FluxPro, and they will give you. They have their own algorithm. It will take some time, and on the basis of that, they will email you with the best 10 or the list of best conformations, best docking conformations that they have documented, and they will send it to you. And Autodoc, as I have said, that I will share with you a detailed outline how you can perform your docking using Autodoc. Okay. Now, these are all where we are getting your lead compound. Suppose you get your lead compounds, it is showing a biological activity as well. But what is important for your lead compound to become a drug-like character is that it needs to pass certain tests. Those tests are since we know those biological processes, means if you see that if you if you just without knowing this all these things, if you want to generally visualize that how a drug should be, it should be, means if you are applying to your body, then if it should be easily absorbed by the your body, so that it can go to its biological target. So it should, the absorption should occur. Now if you want to target, certain different means it can be absorbed from your GI tract, but your target is not in the GI tract, so it needs to be distributed to the region where it needs to function. Okay, then it is also not means it should not hamper the any metabolic process, but it should enhance the process that we are actually looking into. Means for treating a disease we are targeting a receptor for enhancing the biological activity. Also your drug should be stable. If it is metabolized by your system or your body system, then that drug will have no use. So all these things, and if means you also need to consider that how much time your drug is actually present in your body, which will give it means ample amount of time to perform its biological function. If it is excreted as soon as it is injected in your body or it is taken by you, then that drug is of no use. So, and ultimately, which is the most important thing, is that that drug should not be toxic to you. Means you are taking a drug for prevention from a disease, and but if it has certain side effects, a larger side effects, then that drug is not considered to be a novel drug or a good lead compound. You should not consider that lead compound as a drug. Okay. So from these basic understanding, people have came up with certain computationally screening methods. Means this, if you want to test everything of with all those lead compounds, then it will take a huge amount of time, right? So we know this means today in today's field is about all about this data, the data collection, the data processing that we have. There is huge amount of data that is being generated. So for each drugs that is being discovered, so we have certain properties of means properties of the drug that is actually mediating all these processes. So we are actually exploiting the properties of the lead compounds to find the processes, these five processes, which are very important to screen whether your lead compound is a drug like, it is more drug, the druggability is more or it is more drug like, that you need to that you will get only when you have, when you will screen from these properties. Okay. So what is absorption? For a compound to reach a tissue, it usually must be taken into the bloodstream. Okay, so it needs to be absorbed. So there are different ways which can be, it can be absorbed. There can be paracellular transport. Paracellular means between two cells it is moving. Within the with the junctions between the in between two cells there are these junctions, right? These adherence junctions. So through that your drug molecule is moving inside. Another can be transcellular, means through certain endocytosis mechanism, it is moving inside the cell, and between the cell it is moving and doing means endocytosis is happening and the drug is going into the blood. Or with the help of certain transporter, we can also be able to go. Okay, and here also your drug needs to have a cargo. Means certain sometimes this size is so small that or it cannot be. Means sometimes the properties of the drug is such that it is not easily absorbable by your body. So in that time you need to have conjugate your drug with something which can be easily taken by our body, and that can actually help to internalize your drug. Okay, so these all comes under absorption. Then distribution I have already talked about. This absorption, distribution and metabolism, the three makes this pharmacokinetic parameters. Means what is the oral bioavailability, fraction of dose that enters blood in part circulation. What is the absorption, the fraction of dose that passes the gut wall. And what is the clearance, amount of blood cleared per time unit. So all these parameters are falling under this pharmacokinetic parameters, and they are under this absorption, distribution and metabolism. So all this information we will be getting from certain databases. So on the basis of that we are taking a novel compound and see that these chemical properties, if they are present, then it has been previously documented that it is well absorbed or it is well metabolized. So all this information we will be comparing to get this admitox model. Then there is also this absorption is based on membrane penetration as permeation as well, depends on the physiochemical properties of the drug. Just like I said, molecular weight, lipophilicity, that kind of bonding, hydrogen bonding pattern, or the kind of residues that is used for binding to its receptors. Everything is actually dependent for if we want to consider that whether it will be permeable or not. Okay. Then, then we are also going to fix what are the properties affecting the admittance. So these are important because this you need to remember or you need to consider while you are screening your drug likeliness of your lead compound. That is how many hydrogen bond donors or acceptors are present. Sometimes if there are more hydrogen bond donors or acceptors, then it can make also some non-specific interaction which you don't want. So people have came up with different rules that I will be discussing with you as well, Lipinski's rule. So where people have normalized the amount of hydrogen bonds that should be idle in a drug like molecule for humans. So those things are all from the experience as well as from the data collected. Machine learning has played a huge role to in the data curation. Okay, so from that people have found that these are the basic parameters that a drug should follow to become more drug like. Okay, a lead compound should follow to become more drug-like, like hydrogen bond donors and acceptors. Estimation, calculation of hydrogen bonds gives you an idea of how the small molecule, the potential drug, interact with the target as well as other non-specific macromolecular or biological substance that I was talking about. Then the partition coefficient or the distribution coefficient. It is the ratio of the concentration of a compound in a mixture of two immiscible solvents at equilibrium. Normally these organic molecules or the small molecules, they are miscible in an organic solvent and you need to transfer it to a blood. So to blood. So, okay. So there are two solutions will always there for your drug to phase. Okay, so that is the if you take the ratio of the concentration of a compound of in a mixture of two immiscible solvent at equilibrium, that is this calculated as log p. See, it's in a water or in an octane. Okay, so water means most in our blood, it means the most of the part is water. So if you consider the ratio of this concentration in the octanol divided by the in water, then you will get the partition coefficient or the distribution coefficient. And for people have also come up with certain values for partition coefficient that a particular drug should take or small compound should take to be more drug-like. Okay. Now. Then comes the Lipinski's rule for intestinal absorption. Hydrogen bond donors, which are less than 5, the sum of OH and the NH groups. Hydrogen bond acceptors should be less than 10. Okay, so these are all the rules that is been defined by Lipinski. Okay, hydrogen bond donor should be less than 5. The hydrogen bond acceptor should be less than 10. Molecular weight should be less than 500 Dalton. Log P should be 5, less than 5. Okay, the partition coefficient should be less than 5. Then there is something also called as the C log P. Okay, this is the calculated log P, means this is the corrected log P which we actually do it in a which we do it in a means theoretical manner where we know the different means the chemical structure, and on the basis of that there are different functional groups and each functional groups will have certain value, and means we these are all data driven, and previously know that if these functional groups are present, this particular log p value it will take. Okay, so from that we actually also have a corrected log p. This actually helps us to reduce certain noise. Okay, and to more define means, your chemical compound or lead compounds properties. Okay, and there is also another rule given by Warburg where this hydrogen bond donor and acceptor, they should be less than 12, their rotational bond should be less than 10, the total PSA, that means polar surface area, that should be less than 140, the molecular weight should be less than 770. So all these parameters are there. You can just look into these parameters, maybe in means I have seen in your means assignments as well, so you have been asked about these parameters directly. So it's better if you have some idea about the different rules that is being given for this admit property. So this is all that why I want to discuss today, and also I wanted to show you something. So means show you means since we were talking about this admit properties. So one of the thing that I used previously. So I will just try to show you means how you can actually find these admin properties. So there is a web server known as Swiss admin. Okay, so you just search in Google Swiss admin and you will be going to this page which is actually maintained by Swiss Institute of Bioinformatics. So it is very useful to get this your admit properties. Okay, so from here you can actually get the idea whether your lead compound that you are thinking is actually is a how is the druggability score of your lead compound, that can be easily that you can get easily from this Swiss admin. So and as I said that for small molecules, if you want to take the structure, which here you see that they are asking about the smiles. Okay, so what are smiles? So they are the chemical formula of your whole organic compound that is written in means particular pattern. So those are called as the smiles. Okay, so that if you upload, so you just go to PubChem, and for example I want to see means Caspis means there are different capacities, right? Uh, what is that? I actually forgot the name. Um, okay, so just take a certain with any compound. Okay, so for example, this compound, and you that my slides are not visible. Okay, I think multiple times it is actually it went off and it started again. Right now it's visible, right? Yes, so I was just talking about a software, means a web server that you can use, which is known as the Swiss admin. So you can go to the PubChem, which is a repository for the small means organic compounds, and from that you can get your compound of interest. For example, I have just taking for example this compound, and from here you need not to download the structure as well. You just see it is written as isomeric smiles. You just copy this and paste it here. Enter a list of smiles, means you can actually screen a lot of means molecules together. Okay, means by giving different smiles from their means pumpkin, and just click on run. Okay, you see, you are getting a means they are giving a boiled egg. Okay, so this boiled egg is about the different properties they have listed in all these corners of this hexagon. If you see what is the size, what should be how much polar it is insoluble or not. Means all these parameters they have considered, and they have created this region, and if this box that is forming, that is actually abiding this region, then you can say that it is more drug like, means all these parameters is being satisfied for this compound. Okay, so if we can use this for drug designing, then it will be means passing through, it will be it will be then passing through most of the admit test. Okay, then you are also getting the molecular weight here, what are the number of heavy atoms involved, what are the number of aromatic heavy atoms. Okay, so what this is giving you the physiochemical properties. You are also getting the lipophilicity. Sometimes if your drug needs to actually get permeabilized through the membrane, then it should be lipophilic in nature, lipid like loving in nature. So all these parameters, and most what is good about this is you are also getting this total polar surface area, the topological polar surface area. So you just click on these question marks, it will tell you that what this is about, what is the range it should follow. Okay, so all this information you can just get from this software. It will actually use means web server, it will actually help you to get these admin parameters very easily. See here, there all the drug likeliness rules, whether they are abiding or not. So all of these are mentioned here. Lipinski, just we have mentioned now. Okay, then Warburg, what are the rules and whether it is passing all those rules or not. So see Lipinski, it's yes, there is zero violation. It will also give you some idea of whether this molecule is reported previously for any inflammation, any pain, involving any pain. So then this information you can also get, as well as there you can also get the information about the bioavailability, how much it is available in nature. Means if you are designing a compound which is more available in nature, that will actually reduce the cost. Okay, so in this way you can easily get the admit properties. Okay, now, since I have little bit time, so just going means I will share. I will not go in details about Autodoc, but yes, I will share definitely with you this things. This is don't see the slide number because it is a very detailed means how you can perform Autodoc. So I have listed everything here. So I will be sharing with you in this material in the Google Drive, so you can have a look into it, and you can also try installing Autodoc and perform docking. So just I wanted to add some knowledge to you. So these are all the contents, which slide number has which what is the topic about that. So what is Autodoc? Autodoc is a suite of automated docking tools. That's why it is known as Autodoc. It is used for predicting the interaction of ligands with biomacromolecular targets. The motivation for this work arises from problems in the design of bioactive compounds, and in particular the field of computer-aided drug design. Okay, so current distribution of Autodoc consists of two generations of software. So there are means always the modifications, that newer versions are coming. So this Autodoc 4 and Autodoc Vina, there are two major division. Is now one is known as Autodoc, another one is the derived from Autodoc, that is the Autodoc Vina. Okay. So Autodoc 4 has two main programs, one is Autodoc and one is AutoGrid. So as I was telling you, the grid parameters, about the grid parameters you need to specify. So what is Autodoc? It performs the docking of the ligand to a set of grids describing the target of protein, but from where the grids are coming, they are coming from this AutoGrid. It pre-calculates the grids required for docking. So first you need to perform this AutoGrid and then the AutoDock function. Okay, you need to do parallelly, then this docking will work. Then what is Autodoc Vina? So Autodoc Vina is an open source program for doing molecular docking. So I'm not going into the details, you can just read about this in the for your information. So this skipping. So applications of Autodoc, it has Autodoc has applications in X-ray crystallography, structure-based drug design, lead optimization, virtual screening, okay, combinatorial library design. So all this we have talked in some of the discussion, as well as we have also discussed today. So for these are a wide variety of role that you can perform through Autodoc. So there is this Autodoc tools or ADT, which is means if you download the Autodoc, so the graphical user interface is known as the Autodoc tools. Is the free GUI for Autodoc developed by same laboratory that developed Autodoc. Okay, so what are the basic steps that you need to perform in Autodoc? These are these basic steps. So you have the three-dimensional molecule of your receptor. You need to download that. Means then you remove all the hydrogens and add all hydrogens or just non-polar hydrogens. There is an option in the Autodoc where you can add your hydrogens, non-polar hydrogens manually. Assign the partial atomic charges to the ligand, means this is for the receptors as well as for the ligand. In the ligand, you need to assign certain charges. So there are different means atomic charges models which are there. Coleman and Gestiger charges model are there. So that you need to assign first. Then you need to merge all the non-polar hydrogens and their charges with the parent carbon atom. Set up rotatable bonds in the ligand using a graphical version of autotos that you can also skip. This step is not that much important, but then what is important is that you need to form a autogrid parameter file, which has an extension of .gpf. Okay, this is the parameter file that you need to first make before doing the autodoc before doing the docking. Okay, so there should be a grid box, and with the help of this grid box you are creating that file. Then from that you need to convert that file into, since the Autodoc will not recognize that .gft file, that will be recognized by AutoGrid. So for making it recognized for the Autodoc, so you need to convert the GFP file into a GPF file into a DPF file. Okay, and then you need to launch the AutoGrid and Autodoc, and the docking results will be displayed on your screen. So there are different visualization softwares that you can use for visualizing the data of Autodoc, or simply Autodoc tools also you will be able to visualize. Okay, so these are all the processes. I am not going. So the for the for those people who are starting with new with the Autodoc, how you need to download all these processes is given. So means if you also with Autodocs you need to also means download these MGL tools, because many of the packages are linked with this means Autodoc with this MGL tools. So this is also you need to upload, means download. Then this is the interface, Autodoc tools user interface, that it will look like this. Okay, so what are the different components that we have in this? Means this is the main menu bar, gives access to the menus of commands. What are the menu buttons? Okay, so this is the dashboard. Okay, displaying names of the molecules on the viewer window. It also allows some quick actions on these molecules. Okay, so if from here you can actually change. So these all of this means I am not showing you the Autodoc in practical, but yes, but you can change a lot of things from these parameters present. Okay, so this was just means an example that I used while making this presentation. Means we had this means main protease COVID-19 protease, and we wanted to see that this this drug can inhibit that protease or not. So with that, we actually performed is there is already this means inhibitors have been listed in nature. I just checked whether it is working fine or not. So as I said, you go to PubChem, you download your structure. Okay. So this means you get your 3D conformer. So that is in the means you are getting on 3D conformer. The SDF file format for the ligand is available. So you do not have a PDB file format here. So for the ligand, so it is in the SDF file. So there is a software known as OpenBabel, so which you can actually use for converting one extension to another extension. So you can convert .pdb to .sdf to .pdb format in that OpenBabel, and you have your ligand structure in the .pdb format as well. So for this you need to download OpenBabel. So the OpenBabel interface look like this. So where you have to give the input format and the output format. You will have to give the input file. It will automatically in a specified location. It will create an output file. So from SDF you can easily convert it into PDB. Now we also need to build, this is the COVID-19 mail.ts, so we went to PDB and downloaded the file which is in the .pdb version only. So here you don't need to change because it's already in the PDB file. Now, what is suggested is that means for Autodoc, since there are many functions that are involved and many files you need to save, I would suggest you that you make a folder specifically for that means Autodoc, and you save all your files in that same directory. Okay, then the otherwise it has been found that there are been certain errors in Autodoc, and people actually reports that they are not being able to perform docking since they are showing error, because sometimes these file positions is also very important where you are saving those files. Okay, so make a folder and save all the files which are required for your Autodoc in that folder only. So these are all means in there in the Autodoc only you can set a preference where you want to set your all the files, so that can be easily done. Then you need to prepare your macromolecules with the receptor file. So you first upload or they read the molecule into the ADT by means interface. So for that you see this kind of appearance will occur. Then you can also color by atom type. So just looking into the geometries, click on all geometries, then okay. So means all the geometries which are present in your light receptor, they will be shown. Then what you need to do is that you need to delete these hydrogens as well. So and you load module window appears, click OK. So it will actually load a module on which your means Autodoc works. So you need to load the module, and this module will be loaded, and you see the color of your means macromolecule has changed. Okay, so define selection commands that they are not going into that. Then for to select the water molecules with the macromolecules, type HOH in the residue sets entry or in the atom sets entry, click on add. So here you also need to add the water molecules, because you see that they are when they are interacting in the physiological context, so the water molecule will be surrounding, so you will also have the option to add the water molecules in this Autodoc. Okay, and this delete all the hydrogens. Okay, oh sorry, which you are adding, and as well as deleting means you can delete delete the hydrogens as well as add the hydrogens of your means means of orthodox interest. Okay, and these are all means the which chain is actually interacting. We are only we means this information is, as I was talking about, that you need to know about your binding pocket. So here we know that the chain C is only means able to interact with these inhibitors. So for that we just selected those atoms so that we can make a grid box onto it. Okay, so and then we can also we are now means our macromolecule file is ready, and then we are going to the ligand file. Okay, then there we as we said that there the hydrogens and the Coleman charges are being means added. You can also modify your means fine, and also after you have processed your ligand as well as your macromolecule, you now need to save those as a .pdbqt format. So that's why I was telling that there will be multiple files that you need to save for running Autodoc. So that's why we need to save it in a single folder will be good. Okay. Then we are going to the pdbqt file of that means you need to perform a grid parameter file you need to create before to provide the parameters for the grid so that you need to do first. So for that you need to prepare the grid parameter file. Okay, so we have marked the region which are most probable binding site, and we have gone to grid and then grid box. So this kind of box, three-dimensional box will appear where you can actually you can move it in the x, y, and z direction, so that your means the binding pocket is in the center of this box. Okay, then only the docking is more good, I would say. Okay, then you need to save the output as a GPF file. Okay, remember we were telling that auto grid file it will create a grid parameter file. So this is the .gpf file that you are saving. Now you need to start auto grid. Means whenever you will there are two auto dock 4 and auto grid 4. So now you need to run auto grid. So run auto grid. Okay, so it will run and it will give with a file. Okay, this will make a glg file. So there are many extensions which are there in this document, so not going deeper into that. And then you choose your ligand now. Okay, and then your means what field you want to apply for this docking process, that can be also chosen here. And then you have to start the docking. The auto dock needs to be start, and you go to run and run auto dock. Again a file will be written, and from there you are getting this. See, mean binding energy is given, so that you are taking the lowest binding energy. So okay, so this means this is the dlg file. You can also visualize it. Okay, so from these only you can actually visualize. So in the Autodoc tool, so how you can visualize, that can that is also given in this slide. So this is an overall idea how you can perform Autodoc. Okay, so this is all that I wanted to discuss today. And now, if you have any questions, then we can discuss those. Otherwise, I have some practice questions for you. We can just discuss the practice questions and as well as the assignments. And remember, tomorrow at 6 p.m. we will be discussing some questions that I have collected, and so be present, then you will be able to practice. That will be a good practice session for your exams as well. Okay, so Paramita, if you have any question, then you can ask, or I can just directly discuss the practice questions. Okay, okay. So means I will share all these files, and I have also means got some reading materials on this structure-based drug discovery as well as ligand-based drug discovery. For those who are not present, so you can go through the materials. It will be in the Google Drive. So that I don't know how much it will help you in the exam, but definitely if some of you are interested working with drug delivery or drug designing in future, then it will definitely help you. Okay, so now, so if you don't have any question, then we can just go and discuss some practice question based on today's discussion. So these are all the previous year's assignment question. So you can actually take a note down, or you can solve right now, or just take a screenshot. I will definitely share the slides with you. So 2 to 3 minutes you can take, and I have actually 9 questions as practice questions. Then we can discuss the assignments, the week 12 assignments. Okay. Okay, guess you have solved or you have taken a screenshot of this question. So now let us move to the next four questions. So okay. So now let us discuss the answers then. Which of the following are processes of traditional drug discovery? So remember all the in the slide where I have given all the types of traditional drug discovery. So random screening is definitely one. Classical pharmacology is one. Trial and error method is also. So all of these options are correct. So the answer will be D. Which of the following is not involved in rational drug discovery? So, in rational drug discovery, so what are the things that are not followed? The Cuesar modeling is followed in ligand-based drug designing. Pharmacophore development is also being followed. Virtual screening we follow in rational drug discovery as well. But the trial and error method that is more to the traditional drug discovery. The answer will be C. What are descriptors in Cuesar? So on the basis of that means the chemical characteristics of a molecule in numerical form. Means we are considering certain chemical characteristics, we are making it into a numerical form and then applying the function on it to get the biological outcome. Okay, so those chemical descriptors or the chemical characteristics are known as these descriptors in Cuesar. So the answer will be A. Atomic branching and ramification fall in which type of descriptor? So there are this I have not talked about means there are different kind of means descriptors where you means you consider about the atomic means branchings as well as the atomic connections with one another. So that those all fall under this means what is the topology of the molecule that you are looking means topology defines the which is connected how these molecules the atoms are connected in space. So all this information comes from the topology. So the answer is B. So the answer will be B here. What are the physical properties of compounds? So molecular weight, boiling point, solubility, all the above. Okay, so all these are physical properties of a compound. So the answer will be D. What are the basic requirements for molecular docking? So if you remember that means what are the requirements of molecular docking? So you need protein three-dimensional structure as well as library of small molecules. Okay, so the answer will be D here. Which of the following is the docking software? Just I have mentioned, means I have also taught you about how to perform docking in Autodoc. So the answer will be B, Autodoc. What is the docking scenario between receptors and ligands for the lock and key model? So this means uh, for lock and key model. So since it's a specific lock and key, so you need a rigid receptor and a rigid ligand. So for a lock and key model, it is the rigid receptor and the rigid ligand that we all consider mostly when we perform docking. So the answer will be A. Which of the following is present in Lipinski rule of 5? So the hydrogen bond acceptors, if you remember, it should be less than 10. So the answer here will be B. And all others are defying the rule of Lipinski. Okay. Now let us quickly go through the week 12 assignment that is I think the answer is already shared with you. So COMFA COMFA is method is 3D QSR method. Okay, so it is the method used for 3D QSR to set up the models for QSR. Okay, so the answer will be A. Which software is used for multiple alignments of sequences? So the multiple alignments is by a software known as Clustal Omega. Okay, so So there you can do the multiple sequence alignment, which is not the physical property of a compound. So acidity is a chemical property of a compound. So the answer will be D here. Which step is not included in de novo drug designing? So generation of potential primary constants. So if you remember the steps of de novo drug designing, then the primary target constants is not required in de novo drug designing. Okay. So that's why the answer will be C. Which of the following method is used for virtual screening? So all these methods are actually utilized for this virtual screening. Okay, you need to have this admit. You also need to have the Quasar modeling, pharmacophore modeling as well. As for all this, will actually tell you which lead compound is more drug-like. So the answer will be DA. Which of the following approach is considered the ligand-based drug delivery discovery? So the ligands-based drug discovery is pharmacophore development as well as Q-cell modeling. So both A and B is the answer. What are the basic requirements for molecular docking? So library of small molecule, 3D structure of the protein, selection of proper protein ligand complex. All three are required. So the answer is D. Lipinski's rule actually give you your whether your lead compound is more drug-like. So this it tells you about more drug about the drug likeliness of your compound. So the answer is C. Which of the following is incorrect? So the hydrogen bond acceptor should be less than 10. So the answer is B. Which of the following is present in Varber's rule of 5? So if you remember when I discussed the Lipinski's rule, there were also the Varber's rule of 5. So the here all these three are falling under this rule. So the answer is D. Okay, so this is all about today what I wanted to discuss with you. Tomorrow we will meet at 6 p.m. For discussing certain questions that I had actually collected for you to solve. So yes, if you don't have any question, then we can leave for today. Okay, so. No questions, Parvita, then you can leave today and we will meet again tomorrow. And bye. Good night. Oh, okay.